Good morning, my name is Tiffany Amber Onate, and I'm going to talk about the importance of pentose phosphate pathway. The pentose phosphate pathway, PPP, is a fundamental component of cellular metabolism. The PPP is important to maintain carbon homeostasis, to provide precursors for nucleotide and amino acid biosynthesis, to provide reducing molecules for anabolism, and to defeat oxidative stress. The PPP shares reactions with the Intner-Daurauf pathway and the Calvin cycle and divides into an oxidative and non-oxidative branch. The oxidative branch is highly active in most eukaryotes and converts glucose 6-phosphate into carbon dioxide, ribulose 5-phosphate, and NADPH. The latter function is critical to maintain redox balance under stress situations when cells proliferate rapidly, in aging, and for the Warburg effect of cancer cells. The non-oxidative branch instead is virtually ubiquitous and metabolizes the glycolytic intermediates fructose 6-phosphate and the glyceraldehyde 3-phosphate, as well as the pseudoheptulose sugars, yielding ribose 5-phosphate, for the synthesis of nucleic acids and sugar phosphate precursors for the synthesis of amino acid. Whereas the oxidative PPP is considered unidirectional, the non-oxidative branch can supply glycolysis with intermediates derived from ribose 5-phosphate and vice versa, depending on the biochemical demand. These functions require dynamic regulation of the PPP pathway that is achieved through hierarchical interactions between the transcriptome, proteome, and metabolome. Consequently, the biochemistry and regulation of this pathway, while still unresolved in many cases, are archetypal for the dynamics of the metabolic network of the cell.
The oxidative phase. The oxidative weight of this phase comes from the process of oxidation. Oxidation is the breakdown of a molecule as it loses at least one of its electrons. The phase is made up of two irreversible steps. Step 1. Glucose 6-phosphate or G6P is oxidized to form lactone. NADPH is produced as a byproduct of this reaction. NADP is reduced and glucose 6-phosphate is oxidized. Following the oxidation of G6P, another reaction catalyzed by a different enzyme uses water to form 6-phosphoglucanate, the linear product. NADPH has an added phosphate group and is used in the cell to donate its electrons, just like the NADH. Once the NADPH has donated its electrons, it is said to be oxidized. Oxidation, loss of electrons, and is now symbolized as NADP. NADPH is often used in reactions that build molecules and occurs in a high concentration in the cell, so that is readily available for this type of reactions. Step 2. Next, a carbon is removed, cleaved, and CO2 or carbon dioxide is released. Once again, the electrons released from this cleavage is used to reduce NADP. Then this new 5-carbon molecule is called 5-ribulose phosphate. That's it.
Hi, my name is Dylan James Flores, and I'm here to report the non-oxidative phase. What is non-oxidative phase? The non-oxidative phase links the glycolytic pathway to the pentose phosphate pathway, and it allows our cells way to break down ribose molecules ingested in our body. This phase uses several important enzymes, including phosphopentose isomerase and phosphopentose ephemerase, transketolase, and transaldolase. In an oxidative phase, there are 5 steps that we will use, which are the isomerization, transketolase reaction, transaldolase reaction, the second transketolase reaction, and the final step is regeneration of glucose 6-phosphate. In step 1, isomerization. In this step, the ribulose 5-phosphate is then isomerized to ribose 5-phosphate or ephemerized to cellulose 5-phosphate. And then next is step 2, which is transketolase reaction. In this step, transketolase is a thiamine pyrophosphate or TTP enzymes. It transfers 2 carbon unit with keto group from cellulose 5-phosphate to ribose 5-phosphate to form 3 carbon, which is glyceraldehyde 3-phosphate, and 7 carbon, which is pseudoheptulose 7-phosphate. And then next is step 3, which is transaldolase reaction. This reaction involves transfer of 3 carbon unit from pseudoheptulose 7-phosphate to glyceraldehyde 3-phosphate to form fructose 6-phosphate. And next is step 4, which is the second transketolase reaction. In another transketolase reaction, a 2 carbon unit transferred from cellulose 5-phosphate to erythrose 4-phosphate to form fructose 6-phosphate and glyceraldehyde 3-phosphate. And then the final step, step 5, which is regeneration of glucose 6-phosphate. In this step, 2 molecules of glyceraldehyde 3-phosphate formed in step 4 are condensed to form our fructose 6. Fructose 6-phosphate is then converted to glucose 6-phosphate, which is reversal of step 2 of glycolysis, which we will find in oxidative phase.
Hi everyone, I'm Gonzales and I will be discussing about the 2 important products from the process of pentose phosphate pathway. So there are 2. The first one is the ribose 5-phosphate and the next one is the NADPH. So what is the importance of the ribose 5-phosphate? It is actually needed for the synthesis of nucleotides and as well as the nucleic acid, and it is mainly used to make RNA and DNA. And for the next product, which is the NADPH, or the reduced form of nicotinamide adenine dinucleotide phosphate, it is a powerful reducing agent and it acts as an antioxidant, and the main importance of this product is that it provides the reducing equivalents for synthetic reactions such as the fatty acid biosynthesis. So those are the main importance of these 2 products from the process of pentose phosphate pathway. Thank you.